One thing that's neat about this is that it's universal. This genetic code, this flow of information from DNA to RNA to protein, you find it in all life on Earth. And so you can take genes now that we have that technology from entirely different organisms and move them to an organism in which that gene did not originate, and it will make the protein, right? The cells can read those instructions and make those proteins. And so you can take a gene from a firefly and put it in a tobacco plant, and you can get bioluminescence, right? Or similarly, a jellyfish gene in a pig and get bioluminescence. And that is pretty cool. So let's take a closer look at transcription, the first part of gene expression. So it requires an enzyme to make polymers of RNA, of messenger RNA. And so we remember from DNA replication that the polymer that made a new the enzyme that made a new polymer of DNA was called DNA polymerase. Well, the enzyme that makes a polymer of RNA is called RNA polymerase. We also have stretches of DNA called promoters. You find these right before a gene, and they are stretches of DNA that make it easier for the RNA polymerase to bind to the DNA so that it can transcribe the actual gene. And at the end, you have a transcription terminator, right? You have a stretch of DNA that allows the RNA polymerase to sort of bounce off so that you stop transcribing the gene. And between the promoter and the terminator, you get what's called a transcription unit. So we start with a promoter here, right? And the promoter is this little green part. And RNA polymerase is an enzyme is able to bind to the promoter, and it begins unzipping the DNA and adding new nucleotides that are complementary to your template strand, which is the bottom one here, right? And as we add these new nucleotides, 5' to 3', we keep adding new ones here. Eventually we get to the terminator, and we lose this RNA polymerase, falls away, and you get a completed mRNA transcript. And there are 3 stages of transcription. Initiation, which is when we get that RNA polymerase to bind to the DNA. Elongation, which is where you are adding new nucleotides. And termination, which is where the RNA polymerase bounces off. For that first part, often you need chemicals called transcription factors that bind to the RNA polymerase and help it bind to the promoter before the gene. This whole thing is called a transcription initiation complex. One common promoter is called a TATA box, T-A-T-A. You often will find A's and T's in the terminator. This is because whereas G's and C's form 3 hydrogen bonds together, A's and T's only form 2, and so it's easier to hop on and then hop back off again. So TATA box is a promoter. Transcription factors help the RNA polymerase bind to that promoter, and then it can slide down this template strand, make an mRNA transcript until it hits the terminator at the end. So elongation is that whole part of adding new nucleotides on there. The new nucleotides we add are based on the DNA template strand, right? So every time we have a T, we have a A. Every time we have an A, we have a U, because remember, RNA has U's instead of T's. Every time we have a G, we have a C, etc., etc., etc. And termination usually happens at a terminator sequence, which is usually a polyadenylation sequence, meaning lots of A's. And again, A's and T's only form 2 hydrogen bonds, so that the bond isn't quite as tight. So if you have a bunch of A's in a row, we can kind of lose that RNA polymerase, and it's gone, and we're finished making that particular gene. Now, if you are a prokaryote, then that mRNA is finished, right? We just make that primary transcript of the mRNA, and it can immediately go code for a protein in translation. And in fact, it may have already started coding, right? Very often in prokaryotes like bacteria or archaea, the mRNA will have bound to a ribosome before it's even finished being created. Right? But in eukaryotes, it's a little bit different. We have to process the RNA. So first of all, we put caps on either end. So the 5' end gets sort of a modified nucleotide 5' cap there. And then the 3' end gets this poly A tail. So it's a bunch of A's in a row. And those seem to help facilitate the export of the mRNA into the cytoplasm and also protect it from enzymes that might be floating around so that you don't damage the primary transcript. So this is what that looks like, right? We have a 5' cap, which is a version of a guanine here, and then a poly A tail, lots of A's down this way. And then we also splice some of this out. So there are stretches inside the primary mRNA transcript that we've just made called introns that we cut out and get rid of before we make our way to the ribosome. Introns like intruders, right? They're intruding on our mRNA transcript, so we slice them out. Exons will stay there, right? They will be expressed, in other words, right? Exons are stretches of the mRNA which will remain in the mRNA to be expressed as a protein. And this whole process is called splicing. So exon will stay, these introns will be cut out, and then these exons will be put back together. And actually, these exons can be put back together in slightly different ways, right? Not always in the same order, or sometimes you'll have an intron that gets cut out sometimes, but not always. And so I know in the past we've talked about how it's sort of one gene to one polypeptide or one gene to one protein. But really, in eukaryotes where you have mRNA splicing, depending on how you splice that mRNA, you could actually get multiple different proteins from the same gene. And so we only have 20 or 25,000 genes in a human being, but we make way more proteins than that because of this RNA splicing process. So you have these whole contraptions called spliceosomes, which are made up of small nuclear riboproteins, which are these snRNPs, and some other proteins. And they help recognize the parts of the mRNA transcript that are introns and help splice them out. The spliceosome is in charge of that. And so there are RNA stretches involved in this that catalyze these reactions. And so these are RNA enzymes. And RNA molecules that function as enzymes are called ribozymes. And this is a big deal because up until this discovery of these, scientists generally thought that all enzymes were proteins. And this now means that RNA does a bunch of jobs, right? It can not only deliver the information from the DNA, and we'll talk about some other jobs later, but it also can form these three-dimensional structures because it can pair with itself. And so these three-dimensional structures, kind of like proteins, right, can catalyze reactions. Right? Pretty cool. Now, when you get a single gene that can form multiple different polypeptides because you can have differences in how that splicing occurs, right? We call this alternative RNA splicing. Right? So again, the number of proteins that we make is larger actually than the number of genes that we have. All right, so we know transcription is taking the DNA and making a complementary messenger RNA molecule, transcribing the information onto the mRNA so that it can leave the nucleus and eventually go to the ribosome. And we've talked about the splicing that occurs at the end of that production. So after transcription, we have translation. Let's look at that in a little bit more depth. And translation involves going from a nucleotide language like DNA and RNA into an amino acid language. We have to translate this information. And so to translate, you need a bilingual molecule. Well, we have one, and it's called tRNA, or transfer RNA. And transfer RNA is bilingual because it's mostly RNA, but it attaches to an amino acid on one end. And a particular tRNA molecule always will have the same amino acid at one end, right? It only binds to a single amino acid. But it also has at the other end a set of 3 nucleotides, RNA nucleotides, called an anticodon. It's called an anticodon because it will bind to a codon. So if you see this down here, GGC, right, is the codon on this mRNA transcript at the ribosome. It will always bind with a tRNA that has the CCG anticodon because it's complementary. And the CCG anticodon tRNA is always going to be attached to glycine. And in this way, this mRNA codon will always code for the addition of a glycine to our growing amino acid sequence, our growing polypeptide. And this mRNA codon was coded for by a specific DNA triplet code. And so ultimately, there are many steps in between, but the DNA code ultimately dictates what the primary structure of the polypeptide is, right? What the amino acid sequence looks like. So these are three different ways to describe or show the same transfer RNA molecule, right? But either way, we've always got an attachment point for an amino acid at one side, right? This doesn't show the amino acid, but there would be one there. And we always have an anticodon on the other side, right? In this case, it's AAG. And in order to add the amino acid, you need an enzyme, right? This is a chemical reaction. So we have an enzyme called aminoacyl tRNA synthetase, which is long, tough to say word, but essentially it means, hey, I'm an enzyme that takes a particular tRNA molecule and binds it to the appropriate amino acid. And again, a particular tRNA will always bind with the same amino acid. Then we have to take those amino acids from the tRNA molecule and bind them together into a polypeptide or into a protein. And this happens at a different enzyme called a ribosome, right? A ribosome is actually a huge complex of enzymes. It's made of some ribosomal RNA, right, which is another ribozyme, and also some proteins. And there are two subunits. And this is how this looks, right? We have a small subunit and a large subunit, and they will bind together only when you have an mRNA molecule and the appropriate tRNA molecule to begin. Now, on the ribosome, there are really three sites that bind tRNAs, an A and a P and an E. So you can call this APE or EPA, depending on which direction you want to look at it. But really, A is where the tRNA will arrive first, right? A is for arrival, if you want. It's not actually what it's for. It's for aminoacyl tRNA binding site, but I think A for arrival. And then it moves into the next site, P, right, which is where you add your peptide bond, right, when we're going to add this amino acid onto the growing chain. And then E is for exit, right? So arrival, peptide bond, exit. And as soon as one exits, right, the one from the P site will move to the E, the one from the A site will move to the P, etc., etc. Just like transcription, translation also has three phases, and they are also called initiation, elongation, and termination. And they also require some protein factors to get this stuff started. So initiation is where you bring everything together, and it requires a start codon. So the AUG start codon on the mRNA is going to bind with the small subunit, and then the large subunit and the tRNA are going to bind as well. Right? So we start with the mRNA, small subunit, tRNA, then in comes the large subunit, and now we have had initiation occur. And from here, we can go on to elongate, which means to add amino acids to this start amino acid, right? So most proteins, by the way, start with methionine because that's what our start codon codes for. Elongation occurs as other tRNA molecules will come to this A site, and then we'll add their amino acid to this growing chain in the P site, and then this one will move over to the E site and leave. Now, which tRNA can bind here? Well, it depends on which codon we have here. Will it or won't it bind to this anticodon, right? So the codon dictates which tRNA comes, and the tRNA will be carrying a specific amino acid, and so these codons dictate the sequence of amino acids. And we will keep doing this elongation until termination begins. And termination begins when we get to a stop codon. And a stop codon doesn't bind to a tRNA. Instead, it binds to a protein that's called a release factor that kind of breaks the whole thing up, right? So we always start with a start codon, AUG, and then there are three codons that can be stop codons. And they do not bind to a tRNA, they bind to a protein that breaks the whole thing up. And there are some very good videos on transcription and translation that I've posted, and I highly recommend that you watch them, because listening to me yak over some images is not necessarily as good as watching an animation. Now here's an image of the stop codon binding to a release factor, which will break all these components apart, right? The mRNA and the large subunit and the small subunit. And now the mRNA is free to go find another ribosome and make another copy of the exact same protein.